Welcome to my YouTube channel, friends. This is Wiki Chisambo. In this lesson, we want to revise Biology Paper 2, which was written in 2020 under GCE. So this paper is 1 hour 45 minutes, and we want to start with section A. So section A is 44 marks. So you're supposed to answer all the questions in this section. So we have question 1, figure 1.0 shows a plant cell. So this is a plant cell. So when you see here we have B, so this is B, so this is B, and then we have C, and then we have A. And then let us move and see the question. So the question is, name the parts labeled A, B, and C. So we'll go with A. So when you look at, this is a plant cell. So you'll find that here, this is the vacuum. So we would say A is the vacuum. Okay, let us move to B. So when you look at B in a plant cell, this is the nucleus. So this is the nucleus. Okay, let us move to C. So on C, you have to look very carefully. So we have two layers. Number one, this, the outer layer, and then we have the inner. So when you look very closely, you'd find that C is this inside. And what do we call this part? So this is the cell membrane. If it was outside, then that is the cell wall. Okay, so let us move to the next question. Name the organ in the plant in which figure 1.0 is found. Give a reason for your answer. So where can we find this cell? So where can we find this cell? So look at it very carefully. So you'll find that this cell is found in a leaf. So this cell is found in a leaf. So the organ of the plant is a leaf. Okay, so we want to give a reason for or to support the answer. So you'll find that when you look at the leaf or the cell, you'd find that we have these. So we have these. So now these will help us to identify and to know what we are talking about. So we are saying that, so because of that, the reason is the presence, so the presence of chloroplast. Okay, so we move to the next one, to B. So describe two adaptations of the cell in figure 1.0 which enables it to carry out its functions. So adaptation 1. So we can say that adaptation 1, so we can say that when you look at it, when you look at the cell just, you'd find that it has a rectangular shape. So this is a rectangular shape. Now the rectangular shape, it helps to increase its surface area so that photosynthesis can take place. So that is the first one. So we are saying that rectangular So rectangular. Okay, so we can say rectangular shape. Okay. Rectangular shape to increase its surface area. Okay, adaptation number 2, we are saying that presence of many chloroplasts. So when you look at it, you find that we have many chloroplasts. So these are what we're talking about. So many chloroplasts. So that is the adaptation number 2. So we can say presence of many chloroplasts. Okay, so let us move to C. Explain what would happen to the cell in figure 1.0 above if it was placed in a test tube containing distilled water. So what would happen if the cell is now put or placed in a test tube which is containing distilled water? So you find that number 1, the cell will gain or it will absorb water. So when it absorbs water, you find that the cell will become turgid. So we can say that the cell would gain water by osmosis and become turgid. Okay, so we can move to the next question. Okay, so we are done with question 1. So question 2, figure 2.0 below shows the internal structure of a tooth. So internal structure of a tooth. So when you look at it very closely, we have F, and then we have D, and then we have E. Okay, so we can say name the parts labeled D and E in the figure 2.0 above. So we want to know the parts. So labeled D. So this is where D is. So part labeled D. And then we have part labeled E. So the part labeled what? Labeled E. So these are names. So these are names. These are names. Now, where exactly can we find D? So this is the ending of nerves. So we can say nerve ending. Okay, so E. So E, this is the dentine. So this is the dentine. Okay, we move to the next one. So explain what happens to the part labeled F during tooth decay. So part labeled F during tooth decay. So F, so this is F. So what will happen during tooth decay? So we want to explain what will happen during tooth decay. So you find that bacteria will act on food or on food substances that will remain if you haven't brushed your teeth, or if you haven't cleaned your teeth after eating. So you find that because of that, the bacteria will start to feed on those substances. In that situation, they will start releasing acid. And which acid? That acid will dissolve the calcium salt in the enamel formation. So this is what we are talking about. So we are saying that in explaining, so bacteria's acting on food between the teeth, produces acids, which dissolve the calcium salts in the enamel, forming cavities. Okay, let us move. Look at the shape of the tooth in figure 2.0 above. Suggest the role it plays in digestion of food. So when you look at this, so you find that they have the large surface area. So the only teeth or the only tooth that has this, number one, we have the premolars and the molars. They have a surface area for grinding, and then not only grinding but also for crushing. So that is, or those are the roles that the tooth plays. So we are saying that is premolar. So premolar is used to crush and grind food substances. Okay, so we move to the next one. So complete the human dental formula below by filling in the missing number. So we have G, and then we have H, and then we have J. So we have G, and then we have H, and then we have J. So all these, they will make for 32 number of teeth in the mouth. So when you look at G, so we have to find a number. This number, we have to multiply this number by all these numbers so that we can have 32. So which number is that? So we're going to put a 2 here. So G is 2, such that when we multiply, we're going to have incisors, and then canines, and then we have premolars and molars. Now, what about J? H. What about H? So H is 1, and then J is 3. The reason is very simple. Because you should have 3 molars, and then here canines should be 1. So, and then when we multiply, you're going to have 2 on top, and then 2 down. So same applies with all this. So when you multiply and then you add, you're going to find that you're going to be found with 32 teeth. Let us move to question 3. So below shows a section through a nephron. So this is a nephron. So we have J, and then we have K, we have also L. So we have to identify these parts. So when you look at J, J is the glomerulus. So and then what about K? K. So this is what we're talking about. This is K. So this is what we call the Bowman's capsule. So this is the Bowman's capsule. What about L? So this is the last part. This is the last stage, in short. This is the collecting duct. So collecting duct. So let us move. So name the parts labeled J, K, and L. So J, we say that this is glomerulus, and then K, we've said this is the Bowman's capsule. And then we have L. So L is the collecting duct. Okay. Suggest 3 substances that are not filtered out at J. So 3 substances that are not filtered at J. So these 3 here we have substances which comes from, this is the blood capillary. So in the blood capillary we have several substances. So these substances that we are talking about, you'll find that number 1, you'll find we have blood cells, and then number 2 we have white blood cells. So this is red, so red blood cells, and then we have white blood cells, and then we have platelets, and then we can say blood plasma, then we have water, and then we have mineral salts, and then we have glucose, and then what else do we have? Proteins, so all these you'll find, or they will be found in blood. So when you look at here where J is, So J, this is the glomerulus. So here we would find that there will be other substances that will not penetrate, that will not pass through the glomerulus to the Bowman's capsule. So the reason is very simple. You'll find that others will penetrate. So others will penetrate, but others will remain here. Now, those that will remain, number one, they are big such that they cannot penetrate from here. And then those that will penetrate, meaning that they are small, so they will penetrate. So now there are substances which are left here. So now what are these substances? That is the question. So the first one which is not filtered is red blood cells. So red blood cells will remain, and then we have white blood cells also. They will remain, white blood cells. And then the third one, we have plasma. So plasma proteins, or we can say platelets. Also, they will do what? They will remain. Okay, we move. Explain what would happen at J if the coiling of the blood capillary was increased. So the coiling of J. So if it was increased here, so you'd find that it will also increase the process of ultrafiltration. So because of if it was coiled here at J, so you'd find that also here at J, you'd find that also ultrafiltration will do what? Will increase. So we would say it would increase, the process of ultrafiltration. Okay, so let us move to C. Name the hormone that causes the reabsorption of water in the kidney and the organ where it is produced. So the hormone, we want to look at the hormone that helps in reabsorption of water. So you'd find that number one, where hormones are produced, that is where we have to know. So the hormones are produced from the brain, and we have a gland which is pituitary gland. So that is the organ. And then the hormone which is produced there, it is antidiuretic hormone. So which is abbreviated by ADH. And then the name of the organ, so we are saying that it is pituitary gland. Okay, let us move. Okay, let us move to the next question. So the next question, figure 4.0 below shows the external parts of the human eye. So this is the human eye. So we have the M, and then we have N, and then we have O. So we want to know and to identify. So let us move to the question. Name the parts labeled M and N. So parts labeled M. So parts labeled M and N. What are these parts? When you look very closely, you find that M is the pupil, and then N is the sclera. So N is the sclera. Explain how N is adapted to its function. So N, you're looking at how this N adapts to its what? Its function. What is the function of M, by the way? So you find that it performs its function as in to reflect light. So when you look at it, so this one, it is in white, this one, it is in white, and then the pupil is in black. So you find that because of the white color, so it reflects light. So light is reflected, and then black, you'd find that light is what? Is absorbed. So here the function is that white is reflected. So now the question is, explain how N is adapted to its function. So we would say white in color, so it is white in color to reflect light. So we can add to say it is also made of tough connecting or connective tissue so to maintain the shape of the eyeball. Okay, so describe the role played by M and O when a person moves to a room with dim light. So we are looking at a dark place here. So this is a dark room. So a dark room. So now you'd find that M, let us look at M. So M, what is the function? Let us imagine this person is in the dark. What will be the structure of the pupil? And then not only that, what else? But also O. So O, what will happen to O? What will happen to this part, the body? O. Here, what will happen? So what will happen there? So we'll start with M. So we are saying that M, the pupil, so the pupil, dilates to allow more light to enter. So the pupil will become bigger, or it will grow, so that it creates enough surface area for light to enter the eye. And then the other one, we are saying that the iris, so the iris radio, muscles contracts, while the the secular, so the secular muscle will relax. Okay, so explain the cause of short-sightedness and how it can be corrected. So causes of short-sightedness. So the cause is by having abnormal, so by having abnormal or abnormally large eyeballs and having a permanently thick lens. And then correction of this is it can be corrected by the use of concave lenses. Okay, so let us move to the other one, which is genetics. So we have red-green color blindness is a sex-linked characteristic. So red-green color blindness is a sex-linked characteristic. So here we've been asked to define sex-linked characteristic. So we are saying that this sex-linked characteristic is, or we say, these are characteristics. These are characteristics seen as a result of gene carried on the sex chromosomes. Okay, so what can cause sex-linked genetic disorders like the red-green color blindness? So what can cause this? So number one, what can cause this is mutation. So mutation in the gene structure. So mutation in the gene structure can cause color blindness. Okay, let us move. A normal couple had a colorblind son. Use the gene R for normal color vision and gene R small letter for color blindness. Write down the genotypes. So the genotypes of the father and the genotypes of the mother. So you'd find that this is a normal couple. So this is a normal couple. So this normal couple had a colorblind son. So meaning that the gene was passed from one of the parents. Now, because the father is normal, so you find that we write X R Y. So this is a normal father. And then the mother, so we find that because the mother also was normal, so we would say XR and then X small r. So this means that this woman was a carrier for color blindness, and therefore it was passed on to the son. Using a genetic diagram, Show how the son could have been born colorblind. So we want to draw a genetic diagram which carries 5 marks. So the first step that you have to follow is you write parent or parental phenotype. So parental phenotype, so this is normal male. And then this X divides so that we know that we have 2 people. Then so normal mother. Okay, so here we write the second step, which is parental. So parental genotype. So we have this is for the father, who was normal, and then the mother also was normal. Okay, so this is the second. The third one is we write gametes. So gametes, so we separate them. So we write, so this is X, we have R, then okay, so even these we separate them. Okay, so what will follow now is we link them. So this one will go with this one. Okay, so we'd say, so we have a pure breed which is homozygous. XR. Okay, we'll move. So we have XR, then we have Y. Okay, we have XR, we have Y there. So this is what? This is F. So we say F1 of offspring genotype. And then F, so F1 offspring phenotype. Yeah, this is, when you look at this is normal, so we'd say normal female. And then here we have, this is normal as well. She's normal but a carrier of color blindness. So this is normal female, but she's just a carrier. And then we have normal male. So normal male. And then we have a son who was having color blindness. So color blindness male, who is the son. So this is 5 marks. So this is, or this was carrying 4 marks. So this was carrying 4 marks.